Good afternoon, everybody. I will be giving the warm-up talk for not only this afternoon session, but our far evening session many hours from now. So make sure you remember everything that I'll tell you here. It's very important. All right, so this warm-up talk is going to cover three kind of disparate areas, but they're all part of the umbrella of astrobiology. So biosignatures, proteins, and lipids. And as an introduction, if anybody doesn't know me, I'm Bradley Burcar. I'm a NASA postdoctoral fellow working at Georgia Tech. So biosignatures. I don't know everybody's background, so I'll just start real simple. What is a biosignature? It's basically any substance, like an element, an isotope, any sort of fossil that provides scientific evidence that life was once there. And so something that we all hopefully are aware of is fossils from dinosaurs or trilobites or ancient life on Earth that falls in mud, goes under, and it leaves an impression, and you look at it and you clearly go, huh, life was here, fantastic. So it's pretty easy for these large animals, but it becomes a really big problem when you're looking at life as a lot of us are trying to find, where we look at bacterial life, which is much smaller than dinosaurs and starfish. So it is, you can see, smaller than hair. You are trying to find signs of life from before that is this small, so it's very hard to maybe just look at fossils. So what do you do if you're trying to find signs of life earlier on Earth or on Mars or on any other body that you might be investigating? First of all, you have to ask yourself, it's a very important question in biosignature detection, whether or not what you're looking at is a biological product or an abiotic product, something that's just made from like mineralization. Life would be great if it always existed like this, in two just very separate circles. But sadly, it looks more like this, where crystallization and mineralization can leave signatures that look a lot like life, and life can leave signatures that look a lot like crystals. So there is an awful lot of overlap. And this is, in my opinion, the very important fundamental question of biosignature detection, how you can look at a signal, whatever signal you want, and know that it is very distinctively in this green slice, because that is the only way you can definitively prove that life was there, rather than in this ambiguous slice, which it might be really tantalizing, but you have no idea if what you're looking at is from a living organism. So one of the historical ways that people have identified life is through morphological characterizations. And so these are some bacteria samples from about 3.5 billion years ago, from slices of rock from, I believe these ones are from Greenland. And these are the oldest morphological signs of life that we have seen. And you can look at them and they're on the scale of what we would expect bacteria to look like, and they have segmented morphology that is very similar to what we see in modern living organisms. So here's some cyanobacteria comparison. And so we can see cyanobacteria that has left fossils that look very much like this, and we can see that it looks very much like modern cyanobacteria. So one of the biosignatures that you can use is, hey, maybe it looks like life. But as I said, looks can be deceiving, as these are abiotic organic syntheses that can make patterns that are very extremely similar to what I just showed on the previous slide. So you always need some sort of backup information, some more tantalizing info. And so you can look at chemical signatures. And I'm not going to go into the wide breadth of what you can do, but basically, if you have an organism that deposits some chemical and you think you can use that as a biosignature, go forth, try to do it, try to make it happen. And that's what some of the talks today will be talking about, is their specific biosignature that they think is indicative of life. And they'll argue why they think that this is appropriate and see if they can convince you. But morphology plus chemical signatures is a great way to have two lines of evidence in order to back it up. Also, if you're not looking at rock samples or possibly samples that you have in hand, you could be looking for biosignatures spectroscopically. So life gives off chemical signatures. We give off oxygen, we give off methane, as we were talking about yesterday. And so one of the examples that was great from before was if you have ozone and oxygen methane together in an environment, that's a great biosignature because those two should not be able to exist together. So you can look spectroscopically for sources of life, and that's what we're going to do eventually to look at exoplanets. So the main thrust of biosignatures is you want non-ambiguous signals of multiple lines of evidence. This is important because you run the risk of false detections. So in 1996, or actually in 1984, we got this sample of a Martian meteorite. This is one of the most famous rocks that we have in the collection of NASA. So this is ALH84001. And inside of this Martian meteorite is the sample that looks like it's life. And there were some chemical evidence that it was life, and there were, it was pretty popular. In 1996, it was so tantalizing that it was life that President Clinton held a press conference that announced to the world that we had discovered that life had arisen on Mars as well as on Earth. And if you're interested in that press conference, you can actually watch the movie Contact. And in that movie, they play that montage of him actually speaking as part of the movie. But this turned out not to be true, and it made him look crazy and go, aliens. So you've got to be careful before you make really big, humiliating mistakes. All right, biosignatures. Let's move on to proteins. So proteins are a very important part of modern living systems. They do all of the basic heavy lifting biochemistry that we have. And so it would be helpful in a prebiotic world if you had proteins then too that could possibly do biochemistry that's important for nascent living organisms. So how do you get proteins in the first place? Well, you've got to start with amino acids, which are the small building blocks of proteins. And so chemically, we could see that you can mix certain atmospheres in one of the most famous prebiotic chemistry experiments, the Miller-Urey Experiment. They took methane, ammonia, carbon dioxide, and I believe some nitrogen, mixed it together and just sparked some electricity. And they saw amino acids pop out of it. People have spoke earlier of this. This is a very famous experiment from 1952. But you can make amino acids through atmospheric chemistry. Also, through astrochemistry, you can also make amino acids. So here's a piece of the Murchison meteorite, which is a giant rock of carbon that fell in Australia that has been analyzed for its carbon content. And you look inside of it, and you see all of these different amino acids that have been formed from space. So great. We apparently have no problem making amino acids either from space or on Earth. Cool. But how do you string them together? That is a great prebiotic question, which I don't think we are addressing at this conference. But it is a very interesting issue because you can't just mix amino acids together, heat them up, and have them come together. We can talk about that after this talk if you want to, as our lab actually does a lot of work on this. This is the lab I work at. But what we are focused on, if you can find some way to string them together and make these long polymers of amino acids into peptides, how do you fold them into structures that can actually do biochemistry? So this is the tricky part. We need structures that look like this in modern organisms to be able to interact with the other molecules in the environment. But if you just have proteins in the environment, so this is an energy diagram. The lower on the energy is the more thermodynamically favorable product. It's where the molecules will go if you just let them float about for eternity. So their most thermodynamically trapped products are amyloid fibrils and amorphous aggregates. These are non-structural things that just clump together and bind up the proteins into ways that you will have no use for them. When instead we want proteins to be folded into these nice structures that can interact with molecules. And so one of our talks later today will be talking about protein folding on a prebiotic Earth to see how this could happen. And this is a problem because, as I said, the most structures are not the energetically favored products. Once formed, only a very small amount of proteins will even spontaneously fold into any usable structure like beta sheets or alpha helices. So how do you get structurally useful proteins in a prebiotic environment? This is a big question. And lastly, we'll be talking about lipids and the origin of life. And this is important if you believe that the early prebiotic molecules looked very similar to, in some way, to modern molecules. So if you look at a modern cell wall, you have all these proteins and enzymes and other things that are inside the cell wall. But this cell wall itself is made up of phospholipids. So in life, you have these big fatty chains that are hooked onto glycerol with this phosphocholine there. That forms the lipid bilayer that divides the inside of cells from the outside of cells and is a fundamental part of life. So compartmentalizing would have been really useful on a prebiotic Earth too. But this molecule is too complicated to have prebiotically. So we look at much more simple molecules that just have fatty acids without all of these other adornments here. And you can attach them to some other prebiotic molecules that might make them end up to look more like this. And that's what one of the talks we'll be talking about later is how we can get from possibly this to more structures that emulate that to form liposomes, micelles, and bilayer sheets, which can have nice little compartments and areas inside that you can actually do some nice chemistry with. And there's some good animations from the Jack Shostak group in Harvard. They're actually quite cool to demonstrate some of these lipid behaviors. I thought sound worked on this. On the keyboard? Yeah.
Results in the growth of hydrocarbon chains.
Thank you.
That are eventually released from the mineral phase as fatty acids and related compounds. Hydrothermal vents from deep-sea black smolders to land-bound geysers may have been sites where prebiotically important molecules on early Earth were formed. This animation shows the formation of fatty acids deep in the earth below a geyser. Mineral surfaces can catalyze the stepwise formation of hydrocarbon chains from carbon monoxide and hydrogen. Here hydrogen atoms are shown as white spheres, carbon is gray, and oxygen in red. The reaction results in the growth of hydrocarbon chains of various lengths that are eventually released from the mineral phase as fatty acids and related compounds. Because the fatty acids are at low concentrations in the water, they are unable to form higher order structures such as micelles and membranes. Following the violent explosion of the geyser, some water is released into the atmosphere as tiny, microscopic droplets. Fatty acids synthesized along the mineral walls of the geyser are found in low concentration in these droplets, with the longer fatty acids at the air-water interface. A gust of wind evaporates the water molecules in the water droplet, causing the fatty acid to form lightweight airborne particulates that can be transported across the landscape, perhaps eventually settling out and accumulating in localized areas.
You can see that last shot there, how they're forming nice little micelles and vesicles with the nice encapsulated space there from prebiotic chemistry. And that will do it for the warm-up talks for this session. There's no time for questions, so please ask any of the speakers any of the questions that you might have as we go on, or track me down after and I'll answer whatever I can from what I know. All right. Thank you.